All right, welcome back, episode 4. All right, so today I thought what we might do is, obviously in the last videos we've talked about the sort of foundations of this Glycocalyx endothelial surface layer structure, and in the last one we talked about how Nafion provides us an analogue that we can actually study in more detail to understand structure and function. So what I thought we might do today is following on from that, is start to look at, okay, if that is the structure of the Glycocalyx endothelial surface layer, what are the implications of that when we start to look at things like various sort of interventions, if you like? So how does it form? Why does anaesthetic work the way it does? We'll look at things like insulin, diabetes, formation of adipose tissue. Some of the things that we believe this theory of structure of the endothelial surface layer, in particular the glycocalyx, how that can help explain how a lot of these things happen. All right, so that's what we're going to get into today. All right, so I thought we might start with one of the really important papers that we refer to is this, what I call the sea urchin paper, right? The paper that deals with formation of sea urchins, and in particularly the effect of sulfate-free water and the formation, or the availability of essentially heparan sulfate, I suppose. Do you want to talk a little bit about what that paper deals with and why these sulfates are such an important part of early multicellular development?
Yeah, this was something we discovered recently, and it's been around a long time. So the sea urchins are very, very ancient animals, about 700 million years ago, some of the first multi-celled animals, and they weren't developed as embryos from the blastular region, a small number of cells, to the gastropod, where the embryo becomes much bigger and develops into a full animal, without presence of sulfate in the ocean. So it turns out what's missing is heparan sulfate, which is the common polymer that we've seen is the major component of the glycocalyx in our own physiology and all mammalian physiologies. So this polymer is sulfated hydrocarbons, and so you won't get this development beyond this early stage without the sulfate. This is really very interesting, I think, because what it's saying is this is the origin of life, because it forms, you need two things. You need lipids to form a cell, to form an enclosure,
to help containerize
to containerize things.
Yeah.
So you get a multi-layer of lipids, 1 layer, 2 layers, 3 layers, and you get a multi-layer of these polymers, heparan sulfate, that organise in certain ways that we'll come to. So you get the two things coming together, the polymers, the sulfated polymers, which are special, the sulfate's special, because it's one end of something called the Hofmeister series of ions, and the lipids, which are special because they've got to have exactly the right chain length, permeability, and so on. So if you can imagine the sulfated polymers forming this glycocalyx protective endothelial surface layer, and inside you've got this curved multilayer, but the multilayer splits off, and it splits off and it forms little organelles, like the Golgi apparatus and this apparatus. So all the lipids fill in the interior of the cell, and they form these little containerized structures that do different work. So the whole thing goes back to that conjunction of two things, the heparan sulfate type polymers and the lipids together. So that's really quite
So when we talked a little about this in the previous videos, the nature of the lipids matter too, right? It's not just any old lipids, getting the right lipids in the right concentrations of forming. But forming this sort of like sulfonated layer, we can simplify it to that to some degree. This sulfonated layer on the outside seems to be a key component that allows particularly multicellular, right? So I just want to clarify on that formation of the sea urchins is that getting beyond diffuse cells requires this formation of these sulfated polymers encoding the cells. Otherwise, that process sort of stops.
That's right.
Yeah. Okay. So Okay, so we've ended up. I'll try these as my sort of like GC model. So essentially, we can think of these sort of little magnets here as our sulfonated polymers, right? So we have this sort of like rope, right? And what you're sort of suggesting is that because of the chirality related to the again, deuterium playing a role in this as well to sort of spin this up, is we end up with this sort of like these channels forming as these, and obviously, just to be clear, the sulfated polymers in EGC aren't magnetic. I'm just using this as the actual structure to sort of make the point, I suppose. But so if we can form these channels by these.
Do you have a picture, Matt, of the thing?
Yeah, I'll bring that up in a sec,
okay.
right? So I guess what we're talking about here is sort of forming channels sort of like this, right? And that's the, so the advantage of this structure is that it can sort of expand in sort of this direction as the gaps between them, but it can also sort of expand in this direction as well as these sort of channels get a bit bigger. And so, you know, sort of simple, we can start with a relatively tight version like this, or it can sort of spin out to a...
Yeah, the silver ones represent the sulfate.
Yes.
If you like, well, they're one-sided really, and the outside is of the polymers, the cyanic acid hydrocarbons.
But we can end up with an increasingly larger,
small or large.
you know, and again...
and all the channels are connected together.
Yeah, so that's just one channel, and then you can imagine, you know, multiple of these sort of forming that sort of connected network. So it's this change in sort of diameter, and also, so we've got a, you know, a transverse and sort of longitudinal sort of thing. So that's what we're talking about. We're talking about the ability to change that structure in different dimensions, you know, relatively effortlessly, if you like, certainly from an energy sort of perspective, so that we can change essentially things like flow rates and how things. So let's talk a little bit about you know, and the effect of anesthetic and how that how taking different drugs, if you like, or how different drugs can change that structure to affect essentially what we feel, but what ultimately then is things like flow rates of ions, and et cetera, et cetera.
Yeah, fine.
So if we start with something like, you know,
local
anesthetic, right? So, all right, so this is our graphic, if you like, in terms of the glycocalyx. So again, these are the channels through the glycocalyx region, the GC region here, forming these helixes, if you like, that then allow water channels to form in between, and obviously spread out into our endothelial surface layer here, external to the glycocalyx. We're getting this reverse chirality, so we're getting our sulfated head groups on the outside, right? So forming sort of different channels. So what we're talking about here is the expansion of this being to much sort of larger circles, and also expanding in sort of this direction as well. So there's this degree of flexibility with any given channel.
Yeah, that's for the glycocalyx.
For the glycocalyx. And then those channels can then obviously form different junctions as well as a result of that reorganisation of structure.
Yes, but the anaesthetic drugs involve the glycocalyx, those connected channels, but anaesthetics involves the interaction with the membrane.
Yes, right. Yep. So okay, so let's start with drugs then. If we've got different types of drugs, what's going to be the effect of drugs on the glycocalyx structure itself?
Yeah, well, can you go to the multiply connected glycocalyx?
To the...
So those blue ones are your coils of sulfated polymer, the heparan sulfate and other polymers that have the channel through which electrolyte passes, and glucose, and insulin, and all the other molecules that go from the outside of the capillaries into the inside of the cell. But what happens with drugs is the same as happens with the membrane. You can change the curvature, as you just showed, of those connected channels by using molecules that absorb into the surface, the sulfated region, expand the head group area, which changes.
So again, things that basically sort of, you know, going back to this model, sort of essentially spreading it out, right? So
Yeah,
they're
the drugs-
expanding the diameter. We think of it in terms
That's
of
right.
the diameter
Change it,
potentially of
yeah.
the. Yep.
And make it larger.
So you
And
get this
change
versus this, right?
At the same time, they change the connectivity of the channels too.
Yep.
So you're varying the size of the channels and their connectivity. So that affects, with the heart pumping stuff through the capillaries, that affects the rate of flow of the electrolyte, and the rate of flow of the sodium chloride affects the rate of propagation of the nervous impulse. So the drug operates not by actually, how shall I say it? It operates by changing the nature of the glycocalyx, which affects the rate of flow of the blood, and that affects the rate of propagation of the nervous impulse.
And would it be fair to say that, I mean, we're talking about the glycocalyx here as this GCESL structure as if it's essentially ubiquitous. And so I guess it is in the sense that, you know, almost all cells have this, and certainly connected cells are therefore connected. But is it fair to say that there's still going to be a degree of variation in form and structure of glycocalyx in, say, if we look at something like the blood-brain barrier versus, say, in kidneys versus, you know, muscle tissue, etc. So there's going to be some degree of, let's call it natural variation in terms of structure, and therefore different drugs will have different effects in different regions, and therefore this drug affects this region of the body more disproportionately. Would that be fair to say? Yep. Because of that, because it would affect this particular glycocalyx, but this one over here, they're very similar, but they're not going to be exactly the same in sort of all areas of the body. Okay, so you often talk about the sort of the hexane effect of because of the permanency of some. Can we talk about why some drugs are worse than others from their sort of accumulation effect as well?
Yeah. Different drugs some affect the glycocalyx and the flow of blood, and also things like glucose and so on, through changing the structure. But also other drugs affect the structure of the membrane, which adjoins the glycocalyx. And how can we demonstrate that? If we've got a picture of the membrane. Yeah, okay, there. Just look at the lipid bilayer. That's a very simple picture of the bilayer. That's the yellow, very thin 3 nanometer layer of the membrane, which has got the glycocalyx and a very large extension.
So we're talking about this sort of lipid bilayer here. Yep.
So if you're looking at a nerve cell, an axon between nerve cells, the bilayer looks like that schematically, and it's got a lot of structure to it. And the way the nervous impulse propagates along the axon, it opens and closes channels. We've got sodium and potassium channels going through the lipid bilayer, and the rate of sodium propagation affects the signal that's going. But if you have a small anesthetic molecule like hexane is only, well, let's get a dimension. The double chain lipids there look a bit short. They're actually 16 to 18 layers, hydrocarbon molecules thick, like 2 oleic acid, 2 olive oil acid chains together. Each molecule's got 2 chains, and the 2 chains oppose each other. If you put a small hydrocarbon in between the chains, in the middle there of that right-hand layer, the small molecule, like hexane, will interpenetrate in those chains. It's a hydrocarbon, and it opens the chains up. It splits the chains apart, so you can get a wedge-shaped thing. So that presses on. We haven't shown the holes in the bilayer that are the channels there, but the propagation of the nervous impulse involves a phase change of the lipid in the lipid bilayer from what's called a mesh phase. It's got holes in it. The channels open and close and allow sodium and potassium to go in and out. But if you if you change the curvature of the lipid bilayer by inserting a wedge, a small wedge like hexane, it will open it up and it'll stop that phase change of a straight membrane to a membrane with holes in it, and so it turns the propagation of the nervous impulse off, the electrical signal. That's the simplest way of doing it. But for an anaesthetic, one like hydrofluorine, that used to be very common, it's like hexane. Hexane is a simple hydrocarbon. We use octane we use for petrol, which has got 8 hydrocarbons and nothing else. But if we use something like hydrofluorine, it's got, instead of hydrocarbon, it's got fluorocarbons. And the fluorine molecules are CF2, 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 where for fluorine. So if the fluorine molecules are much bigger, much more hydrophobic, they go into the lipid bilayer and have a big effect that causes the anaesthetic effect, switches off the phase change that propagates the nervous impulse.
So, sorry, just to clarify, so what you're saying here is that, so this hexane, because it's so small, it can bury itself in here and...
So can hydrofluorine.
Yeah, sorry, but it buries itself in there, and so it can sort of sit in that layer, and because it's sort of, I guess, you know, push the layer out, that's what's having the effect, essentially.
So, it changes its curvature.
And so the
But fluorocarbon is so hydrophobic that it comes out.
Yes, okay.
If you have fluorine gas and you breathe it, it goes into the brain, it changes the membrane, switches it off, or the mechanism I was talking about, but then it leaks out. But with a hydrocarbon like hexane, which you can take in with petrol sniffing, it goes in there, it won't come out.
Yeah, okay.
So you do permanent brain damage.
Because you're essentially the more you do it, the more you're accumulating, whereas if you have the fluorocarbons, you can take it now, it'll leak out later, and then you can replace and continue that dose.
And you can do that in a very simple experiment. If you take lipid bilayers in what's called liposome, multiple layers of lipids, a white suspension of liposomes in water, if you bubble in hydrofluorine, you can see it after about 5 or 10 minutes. It suddenly switches to a different state, what's called a cubic phase, which is...
Because of those packing constraints.
It's clear instead of white, and it changes the structure, and you can see the structure changes into a bicontinuous structure, which is the 3-dimensional analogue of the 2-dimensional phase change. And then if you cut off the bubbling in, out it comes and it switches off. So it confirms the mechanism.
So by cutting off the supply, as it leaks out, it doesn't get replaced essentially, and therefore it goes back to that chain. So again, it's a mechanism that goes backwards and forwards, whereas with the hexane, because it doesn't come out, once you've accumulated the threshold, you're basically stuck, literally stuck.
Your brain is stuck forever.
Yep. Okay. All right. So without going into the details of, I mean, we can, the million different examples of different drugs, etc. But I guess the point of this that we want to sort of get across is the fact that you've got 2 different types of mechanisms that can affect what you would call as the effect of a drug, you know, whatever that drug is that it can affect the curvature of the glycocalyx, and it can do that either temporarily, you know, in a minor way or significant way, and it can do that sort of permanently if it was able, you know, if it is enough to sort of disrupt the because the glycocalyx itself, would it be fair to say that because the glycocalyx is relatively dynamically stable, it relatively is, and some of the work Hans Vink's done in terms of showing, you know, disruption of the glycocalyx and its ability to sort of repair itself, it's a lot more dynamic than, say, the cell membrane would be. If you once you reach a threshold with a cell membrane and wreck it, you're pretty much killing the cell, right? Whereas the glycocalyx has a bit more tolerance, would you say, in terms of its ability to reform?
Well, there's 2 things. Broadly speaking, what you're saying is right. Well, I think it's right. But what we're talking about with the hexane and hydrofluorine is anaesthetic agents, and there are many. It's affecting the lipid bilayer. If you have what should we call it, an agent like Cepacol that you use for colds and flus, you'll take this cationic positively charged lipid or surfactant, and you swallow it and you take it, it gets into the membrane too, the same as hexane does. It absorbs into the membrane, but when it absorbs enough, it breaks the membrane open instead of changing the curvature slightly. It breaks it up dramatically, and that destroys the cell membrane. Okay, so that's a dramatic, that's what you do when you take an agent like Cepacol for colds and flus. So that's one thing, the detergent action.
So the way it kills the bugs, if you like, is it's rupturing the cell membrane and therefore...
kills the bacteria.
Yep.
But in lower concentrations, it will go into the membrane again, and then it behaves as partly an anesthetic, but also at low concentrations it behaves as a immunosuppressant, and the immunosuppressant is a big factor. So you have these several factors all together, but they're all kinds of reagents that can affect these changes. There's one effect on the membrane, the detergent action, destroy it. The other one is on changing the nervous impulse by changing the curvature. And the other is an immunosuppressant action, which changes the nature of proteins in the cell membrane. And then with the glycocalyx, you have similar effects that change the phase of the glycocalyx, the diameter of its connected tubes, and its connectivity, so it changes the flow rate. Many, many factors can change these things. It's not just one drug or the other. Yep. There are many things that do it, and that's an important issue to say. That.
And the advantage of that means that therefore it is very dynamic, but the disadvantage of that means there's many things that can sort of...
Well, that can happen with the cell membrane too. Yep. But you can, a drug is sodium chloride or potassium chloride. You can change the diameter of these tubes we're talking about and their connectivity by using different salts or different proportions of salts. Yep. So you can collapse the whole endothelial surface layer if you have too much salt, too little salt, and then everything goes haywire.
And there's some papers we refer to that have shown that as well. And again, going back to Vink's work, using heparanase, etc., to show that the collapsing of the glycocalyx, you get things like your leakage of cholesterol and your inflammatory responses with the immune system as well. But
I'd say what we want to say, I think, is that there's not just one magic drug that does things. Yeah.
And the dose is the poison too, right? Because I think what you're saying there is a small amount might not. And I guess the distinction I'm trying to sort of allude to here is the cell membrane versus the glycocalyx is if you're destroying cell membranes, then you're going to get a pretty acute specific response, right? Whereas you can mess with the glycocalyx more readily, more significantly, without necessarily getting symptoms you might sort of recognise, right? If you're killing cells, you'll generally get some sort of measurable response from a symptom perspective. So you'll know that too much of that drug is causing these problems relatively quickly.
Yeah. The catastrophic side, if you're destroying cell membranes, if you break the membrane, then you're going to release an enormous flood of potassium because the inside of the membrane is all potassium, 98% membrane. The outside in the blood is all sodium. So 98% sodium outside, 98% potassium inside. If you break the membrane, a cell is going to release a huge amount of potassium, which is going to have enormous effects outside. If you collapse the endothelial surface layer by using a different salt concentration, it collapses, it stops the flow of nitric oxide, of oxygen bubbles, stops the flow of carbon dioxide. And so, again, catastrophic. So.
But would it be fair to say, though, or would it be a reasonable assumption, to say that at least if you're destroying cell membranes, you're likely to realise that, from a physiological perspective, you're likely to realise that relatively acutely, right? Whereas you could be continually having a substandard, and this is, we talk about atherosclerosis, but you can have a continuing degradation of your glycocalyx in certain areas of the body for a very long time before you really realise, because cells dying, you're going to get that. You're going to get once you reach a threshold, you're going to get a noticeable rise in some sort of symptoms, right? whereas a small or progressive degradation of the glycocalyx isn't necessarily going to show up symptomatically in the same way.
No, your metabolism will slow down. You won't. You'll with the endothelial surface layer, if it's broken, you're going to have deposition of lipoproteins and other crud on the surface of your capillaries, and you're going to have heart disease because of crud.
And so that's essentially part of the aging effect, if you like, is the progressive degradation of your glycocalyx over your lifetime. We would suggest is going to have a significant impact on your relative aging capacity.
Yeah, it affects aging capacity, but it can be affected by different illnesses too.
Sure, 100%. Yeah. And so it's, I guess the part of the point there is it's not just the ability to maintain that structural integrity of your Gs through your lifetime is going to be critically important, and we'll talk a bit more when we talk about diet and exercise, but the ability to provide the resources required to keep your GC healthy is obviously a key part of what allows, and we'll talk a little bit about that in particularly in terms of diets, but making sure that the right balance of the right constituents are there, you know, your sulfated polymers, the right salt concentrations, particularly your trace elements, etc., have a huge impact, because as you say, if you're continually flooding your system with lots of the wrong salts, you know, because, you know, cell membranes have been rupturing in this flood of potassium, for example, then you're going to have a continual degradation of your glycocalyx. Okay, so all right, so let's move on to sort of the whole insulin sensitivity, diabetes, adipose tissue formation, because I think this is a really interesting story, and I think this structure provides some really good insights as to maybe what is actually happening in terms of how different people can have, you know, how different people can have different levels of insulin sensitivity, and even how the same person can have different states of what's referred to as insulin sensitivity, right? So this idea of insulin sensitivity being for a given level of insulin, I seem to be able to clear more or less glucose effectively, because diabetes is really, you can think of diabetes as essentially too much glucose in the blood, right? So it's a glucose poisoning effect. And that can be because I'm just eating too many things that quickly produce glucose. So, you know, very high glycemic index orientated food, for example, or just consuming too many calories all up. Or it can be the fact that I don't have enough insulin, or the insulin's not doing its job properly. They're the sort of two ways you can end up with too much glucose in your blood. It's not being cleared out. So under this sort of model, what we're sort of suggesting is, again, if I go back to if we've got really sort of essentially tight channels, then your flow rate's going to be different. So we talked a little bit previously, but I want to go into a bit more detail of how the insulin sensitivity works from opening up those channels perspective. So your insulin molecule it's hydrophilic, so it's going to sort of come down these sort of water channels. And then you just want to talk about how that then expands those channels?
Well, we think, we know from a bit of various studies that insulin goes into those channels as a linear molecule. Mm-hmm. When it's flowing in the blood, it's coiled up as a protein, but it opens up and snakes into those channels. So it can
open out into a big chain, into a big long string, and then...
Yeah, it goes into the channels and will affect the curvature and will affect the diameter and thickness. So it opens them up.
So it basically takes one that's thing and pushes that channel out, and it does that by essentially changing the hydration forces on these sulfonated head groups, yeah? Yep.
Yeah, we think so. And then, so it allows passage of the glucose through.
Yep. Well, it opens up the channel, right? So therefore, the bigger channel is more glucose, because the glucose is essentially using that aqueous channel as its primary transport mechanism into the cell. That's at least the theory, right? So in other words, if insulin... So you need a certain amount of insulin, right? So obviously you don't have insulin because you're type 1 diabetic, that's a whole different story. But if insulin's there in sufficient quantity, then in theory these channels are going to be sort of opened up and allow good insulin flow, and therefore we're going to have what we call a high rate of insulin sensitivity in that sense.
Well, it's more complicated than that insofar as the whole glycocalyx endothelial surface layer. There's a whole bunch of porous media that allows oxygen, nitrogen nanobubbles. Mm-hmm. that are circulating in the blood to come in. They're involved in producing activated oxygen species and nitric oxide, a whole bunch of different enzymatic reactions. And they're going into the cell as well through the glycocalyx. And they're nanobubbles and they need to be assisted to get through, to have a channel to get through. And I think this is coupled to the peculiar bubble-bubble interaction problem. If you may recall from the further discussions that if you have bubbles coming together, they won't fuse for some salts, and they do fuse for others. If you have sugars, if you have bubbles coming together, glucose has no effect on the fusion of bubbles, and fructose and sucrose do. So they have different transport properties involved with gas. Depending on the sugar concentration, glucose will go one way through these hydrophilic channels, the sulfated channels, and fructose and sugar and other sugars will go on the outside of the channels, which is all a porous. It's a hydrated sugar region and sialic acid regions. They're all sugar-like molecules on the outside of the sulfated channel. So it's coupled to the whole transport of gas system as well. So we've got the glucose, we started with the glucose going in. I'm saying it's a more complicated.
There's a lot going on. It's not just motorcycles on the freeway. There's 18-wheelers and okay. So, but just if we park the buses and the, and the utes and SUVs and everything aside, just for to keep it as simple as possible, at least from a starting point, and we can expand back out in complexity. So it sounds to me like what you're referring to is so we have these channels. These channels can change in diameter in particular, we're referring to predominantly here, right?
And connectivity, because...
But the insulin's changing that structure so that the diameter changes and therefore also the number of connections, etc., change.
And I'd add to that what we think too is insulin's called a hormone.
Yes, yeah.
All hormones behave the same way. That is, they're not actively involved like an enzyme.
They're not going and doing a specific job. They're changing something that allows that job to occur.
They're changing a membrane or they're changing a lung surfactant. Yep. They're changing an intricate structure which is there for a particular purpose. So if it's damaged, the hormone comes along and fills in for a missing protein or whatever. But it does so physically. It's not doing it with an actual reaction. Insulin's not interacting with
Insulin's not doing a chemical reaction with something to create a result. It's creating a physical reaction, a physical changing structure in some physical way, which allows things to occur.
Hormones behave like that, not like enzymes. That's the difference. They're a physical change in nanostructure. That's
okay, and and part of the potential evidence for that is I go back to insulin sensitivity. A certain amount of insulin can have a varying effect, and in fact, we know that if someone's suffering, if you like, from insulin sensitivity or diabetes, giving them more insulin isn't going to change the effect beyond a certain threshold, right? So if someone suffering from type 1 diabetes, they literally don't have insulin, then insulin injections are going to help because it's going to have this effect. But if I have sufficient insulin, adding more insulin isn't going to change its effect because something's stopped either that these channels have expanded as far as they can. And
also the hydrophobicity changes, the hydration of these sulfated tube changes. And because the different hydration affects how a molecule can get through.
In terms of the transmission, okay. So, but let me just start with the simplest version first, right? So I look at this and say the channels can get larger and therefore affect flow rate, but that means that the structure itself has to have the flexibility to get that way. There has to be sufficient, heparan sulfate, chondroitin sulfate, the other conditions, things like SOL conditions, the other specific ion effects. You alluded to earlier, there's a whole mess of things going on here. So it's quite possible that the right amount of insulin still can't expand these channels sufficiently because of pressures from the inside or the outside are counteracting the forces essentially that this insulin's trying to do. So the insulin's trying to expand the channels, but because of other things, and we'll talk a bit about glucose leakage in particular, there can be other factors external to that helix in the hydrophobic areas out here that essentially sort of keeping that channel compressed.
Too much insulin will change the surface of the channels, and that's the water structure.
Yep.
And so that affects the transport too.
Sure. But I guess what I'm alluding to is completely independent of the level of insulin. Okay, there can be other things, and things like, salt concentrations being a good example, that prevent these channels expanding in the way the insulin is trying to get them to expand, for want of a better description.
Well, but also if you've got too much insulin, it will partition into a different part of the glycocalyx.
Yeah, okay.
In the same way that
Yep.
the insulin's coming along
Yep.
From the liver and in the capillaries and flowing along, it's partitioning from the blood into the channels.
Yeah, okay. Yep.
And similarly, if you've got too much of it coming through,
It's got to go somewhere.
Into the outside
Yep.
of the glycocalyx,
Yep.
which is still hydrophilic,
Yes.
but it's all a sugar.
Yeah, I think that's a good point, because I guess, again, keeping in mind that these things themselves, they're not PVC tubes, right? They're not...
How do you know?
Well, they're not PVC. I guess there's a degree of porosity, I suppose, is that things can move in between these helixes, right? So they're not... Yeah. So things can be going, in between here, in and out as well. So I guess the point I'm trying to get here is insulin's primary job, if you like, in terms of this model, is it's to expand these channels. It will do that if it's there in sufficient quantities, and therefore allow a certain amount of glucose through these channels. Okay, let's call that normal function. But if the other conditions aren't there because of various dietary or whatever factors, particularly salt concentrations,
gas coming in, breathing, and stuff.
You're not doing sufficient exercise. We'll talk about that in a second. But I guess without looking at, there's a whole bunch of different conditions that can prevent the insulin from doing its job that have nothing to do with the amount of glucose and have nothing to do with the amount of insulin, right? Is that could prevent these channels, or in fact these channels could expand too much. Either way, there's a limitation on how much glucose is going to get through this channel, regardless of how insulin's done its job. So in any particular circumstance, whether I'm healthy or unhealthy, the channel can only get so big. So if I then overload the amount of glucose, and then there's only so much glucose that can flow through this channel, the excess glucose is going to leak into, if you like, for want of a better term, because it's all sort of coming through here, right? It's going to, rather than go down through sort of these in through these water channels, it's going to leak into, if you like, the more hydrophobic area, if you like, in between all of these channels. Yeah.
It's not really hydrophobic, but it's less,
relatively, I guess.
It's less hydrophobic. Yeah, it'll go into the outside of the sulfated.
Okay, and if we so to take that a step further, then we have all this glucose sort of leaking into this extracellular area around the glycocalyx. This is where the formation of the triglycerides now come along, and this concept of the glycocalyx is a bit of a reactor, if you like, or a factory, for want of a better description. You get the formation of triglycerides and ultimately formation of your adipose tissue as a result of what do we do with all this excess glucose, because it's not going to make its way into the cell.
Yeah, well, we think so, because the conventional idea is that the glycogen, or the very complicated molecule that gets incorporated into the adipose tissue forming cell, is a big molecule that's manufactured inside the cell, we think. Well, that's the classical view, but it's a great big molecule, and it's very difficult to imagine it getting through that membrane.
And in fact, we referred to some papers that suggest otherwise, right? That the formation of adipose tissue is an extracellular process.
It's an extracellular region, and the incorporation of the triglycerides into this adipose tissue cell, and the formation of the multiple sugar big molecule for them is done within the glycocalyx itself.
Yes. Yep. And again, I don't want to go into detail of this one because it's probably a bit too much of a rabbit hole for today, but there's also some evidence of the role of heparan sulfate in terms of mediating that formation of adipose tissue as well. We've got some references in terms of that. So there seems to be a lot of evidence supporting this idea that glucose, excess glucose, whether that's because your insulin sensitivity is very, very low and therefore the channels are quite narrow, there's not a lot of glucose getting through. So despite the fact that you're not spiking your glucose, that you're not overeating, for example, but your insulin sensitivity is so low, is that still the channels aren't large enough and therefore you can still spill into this glycocalyx area and form adipose tissue even though you're not overeating. Or alternatively, even if you are forming these channels and these channels are working as effectively as they possibly can, you can still overeat essentially. You can still spike too much glucose production.
Well, there's also a balance between the insulin and the opposing molecule that's issued by the liver, the glycogen. Yep. So it can also...
The balance of the channels, yeah. I guess, again, so there's lots of ways that nature, the body, however you want to describe it, has created ways of mediating how big a channel do we need, and obviously a lot more going on than just the insulin glucose story, but you can start to see here, hopefully we're sort of forming this story of saying, look, glucose can go in or outside of these channels, and how much of it goes inside or outside is going to be a determiner of how much adipose tissue is formed for any given level. And you can think about that from an evolutionary biology perspective, as that makes perfect sense, that we adapt a storage mechanism for energy that is, again, very, very simple, very dynamic, and can be done acutely in different areas of the body, so that we have this. If we can't utilize that glucose straight away, let's store it nice and effectively and efficiently by changing the nature of these channels and therefore how much is easily consumed by the cells.
Not quite so simple.
Well, relatively, right? It is. So
this is dissolved gas too, which is also playing a role, which is coupled too.
Sure, but I guess in terms of trying to get the relatively simplistic message across of what happens to glucose and how does adipose tissue form, there's an almost infinite degree of complexity that changes all of those things. We can accept that from the salt concentrations, even things like, we haven't spoken about temperature yet, those sort of things, but there's lots of things that can affect that. But in a normally functioning body, we can now hopefully start to see how relatively small changes in insulin can have an effect or not. Very large changes of insulin levels can have an effect or not, because they can be so easily counteracted by so many other factors. Yeah, and so people can form adipose tissue despite the fact that it seems like they're healthy, right? People can not form adipose tissue despite the fact that it seems like they're not healthy, because again, there's so many different things that can change the nature and structure of these channels dynamically that that you can get this varying range of degrees of sensitivity. Would that be fair to say?
Yeah.
Yeah. Okay. All right. So you also talked a little bit about previously around the effect of, and I know when you talk about vision and the way to sort of, particularly the importance of these sulfonated polymers in the formation of eyes, rods and cones, and how those structures are formed very early on in the development cycle of an organism. Do you want to talk about how you talked a little bit about last time in terms of the ability to sort of channel switch sort of significantly, but what role do you think these sulfonated polymers play in that overall connectivity to allow all of these different cell functions to cooperate, if you like?
You mean in vision?
In vision, yeah.
Well, I don't know if we discussed it before, but as far as I can see, that we know that in the vitreous humour of the eye, the clear fluid through which light goes through the lens and into the back of the retina, it's all stitched together by thin strings of polymers, which are all heparan, chondrite and sulfate. So clearly it's connected to the glycocalyx, because those things are teased out of a glycocalyx, which is made up of those polymers, just like the endothelial surface layer. So it forms the gel through which the light goes. And also, at the same time, it's got a lot of hydrolurin, the eye, as a lubricant around, which is also connected to it. So the gel that forms the eye, the bulk medium, seems to be an extension of the endothelial surface layer, and it well that connects it through the rods and cones, which are stacks, folded stacks of lipids, essentially, and they can switch from one form to another in the same way that we had the switch with anesthetics. The lipid bilayer can switch to one with holes in it, through which you've got sodium and potassium channels. But in the bulk system, like another system called the mitochondrion, you have this bulk, say it's stacks of these rods, okay, folded bilayers. But they can switch when you have light coming in. It's a lipid, mainly lipid region. Light comes in. It hits a molecule called rhodopsin, which is embedded in these stacks of lipids. And that causes, that absorbs the energy, and that causes the phase change of this multiple bilayer rod or cone to what's called a cubic phase, opens the channels, so you get this bicontinuous structure, and that allows the passage of sodium and chloride through the endothelial surface layer surrounding this thing. And so this tracks, it gives a spike in the conductivity and triggers off the nervous impulse at the back of the rods and cones. So they're connected together in the same way that all the other ones are.
Yeah, so this is the genesis of the signal, if you like, of what we see is coming because of this reaction of the photon. And again, so going back to our sort of sea urchin discussion earlier, the requirement for these sulfates to allow these structures to form early on in not just multicellular development, but the eyes are one of the very early organs, if you like, that are formed as you know, as we grow, if you like, from zygote on, sort of thing, and it's a teasing out of these sulfonated structures.
Yeah, it's a rearrangement of the endothelial surface layer, cell multiple bilayer system. So yeah, that makes sense. The photon that strikes the things and triggers them off, they're delicately poised, just like the drugs and the glucose and everything. They can switch from one form to another. But to be able to switch, you have to have the concept that the way these structures are arranged, they're bicontinuous, and they can switch from like multiple bilayers of lipids that are impermeable to open channels to bicontinuous. And well, you need that concept, but once you see it, then it all seems to fit together.
And it's quite the mental leap to think that a house of cards, if you like, with a flick completely rearranges itself to an entirely different structure, from a but this mechanism is all through all of these biological structures, and
including mitochondria too.
Yeah, but the idea that it's it goes from what seems like a relatively static permanent structure to another completely different seemingly static permanent structure, because when we look at images of different look at images of these different structures, it can lead us to think, or at least give the illusion of these structures themselves are semi-permanent, because when we look at structures in the macro world, we don't see a building suddenly turn from a parking garage into an office tower sort of thing.
Or me, no, I'm just a permanent structure.
So we have this idea at the macro level that things have this degree of permanency and rigidity, if you like, that is simply not true when we get down to the...
Yeah, a microscopy captures an image, it doesn't capture the dynamism of the whole system.
Yes.
I mean, we discussed before that lipids in the membrane, the flip-flop rate of a lipid in a bilayer from one side to another is 3 months.
Yes, yeah, okay. Yeah.
So that they're pretty static kinds of things.
Yes.
On the other hand, with the glycocalyx or with propagation of the nervous impulse, they go very fast. opening and closing channels. So it's fixed, but it can open and close at the same time.
Yeah, yeah. And we talked about the idea of, like a strobe light on the dance floor sort of thing, as opposed to just a photo of a building. Yep, yep. Okay, so let's just go a little bit further then and look at the GLP-1 drugs, your Ozempics and those sorts of things. Why are they having why is it you think then, looking at this ESL GC structure, why do you think they're having such a huge impact on the way people are essentially processing sugars and therefore fat tissue?
Because we've got an excess of this large molecule, this sugar molecule, which is formed outside the connected tubes of the glycocalyx, the sulfated water channel, and that we've taken to be producing adipose tissue. It's producing these sugars. So the Ozempic is a similar sized molecule, and it can't take place in the metabolism, but it goes into the same region and displaces the glycogen and the other molecules. So it stops the adipose tissue forming.
Yeah.
That's simple.
Yeah. It just basically just gets in there and shuts down the ability for this triglyceride to adipose tissue formation scenario. Yeah.
Well, it seems to be in the same way that hexane stops propagation of the nervous impulse.
So it's, again, maybe a little bit overly simplistic, but essentially what it's doing is it's disrupting the natural progression of the accumulation of these products in the external region of the glycocalyx, the outside channels, if you like.
And so, and by the same token, it can also wreak havoc in the whole metabolism of the whole system. It's very delicate. And so you get extreme, you can get extreme reactions to the thing.
Yes, yeah. And so would it be, and maybe this is getting sort of a bit sort of speculatively, but the impact of these drugs seems to be essentially on appetite suppression, right? It seems to be because I don't feel as hungry as often, I eat less, and therefore I reduce my calories, and that's the effect of it. But would it be reasonable to speculate at least that it's almost like a signal of the body saying, hey, we've stored enough adipose tissue for now, we don't need to sort of keep eating because of it's interrupted that, but it's, I guess, sending a similar sort of signal, would that be a reasonable?
I guess so, but from a physical point of view, it must just go into the glycocalyx in that region outside the water channels, and it fills in for the naturally excess glycogen, or whatever the name of the molecule is, and it replaces it. That's it. So, so the adipose, you can't build the adipose cells anyways.
Yes, because it's interrupting that process. Yeah. Okay, all right. So we might just finish off then with just a couple of other sort of related things. I would talk about the effect of exercise. You talked about, particularly the nanobubble formation and transmission, the requirement of that sort of thing. So, in terms of the effect of exercise being essentially, the more physical activity we're doing, or obviously therefore ingesting more O2 and N2, we're breathing a lot more, we're producing much more metabolites, in particular CO2, and therefore there's a lot more, would it be fair to say, a lot more passage of these nanobubbles through that structure, which helps keep the structure sort of functioning properly. Would that be a reasonable summary?
Yeah, and the most impressive evidence for that is recent work of Russian colleagues who have looked at, and other people, looked at shaking of water, shaking of liquids, raindrops falling. If you look at raindrops falling, you find that when they hit a hard surface, it's like banging the ground when you're doing running and so on. It produces activated oxygen species, and nitric oxide and others. So all of the activated species that are formed with nanobubbles are formed because of the nanobubbles, because of induced by the shaking, by the exercise. So that actually increases the metabolism, the availability of activated species.
So it's not sorry. So just sounds to me like what you're saying there is not just the fact that I'm breathing a lot more, right? So I'm emptying in my lungs and bringing more in, but you're also talking about the physical activity, the vibration, if you like, of that. Because I'm running or I'm doing something that's sort of got me moving out.
The banging, the vibration increases the reactivity because there's heaps more of these activated species hanging around. And you can see that if you lay on your back in a hospital bed for 6 weeks, you lose 25% of your bone mass because bones are continually repaired and built. So you've lost it all because the exercise is not there. Excuse me, the activated species are not there.
So not just the fact that you're carrying the weight around as well, but the shaking. And again, you look at some of those remedial activities or devices that I know are used. You sort of sit your feet on the vibrating kind of platform, etc., and they're good for circulation and those sorts of things as well. There's obviously that's further support to that. The idea that the more physical movement you're doing, not just from a respiration perspective, but the fact that you're, as you say, you're sort of shaking up and creating more reactivity within the system as well.
That's a good theory,
So because I guess that helps to explain or at least give some sort of insight into why different types of exercise, really does work. And it's not just a case of controlling your calorie intake will make you healthy. Controlling your calorie intake can make you form less adipose tissue, obviously, but the reason why the VO2 max, I know, for example, is very highly linked to essentially longevity, not just how long we live, but certainly the quality of how we live. VO2 max, the ability to consume oxygen, right? So have people's ability to, the amount of exercise they can do, so think of it as sort of like, lung capacity, for want of a better term. So there's some other implications, though, in terms of looking at this structure. In particular, I want to talk about sort of hibernation, right? And so how are some creatures able to, how do they know, that it's spring and it's time to wake up? How do they know that it's winter and it's therefore time to sort of, because hibernation isn't napping, it's not having a big sleep. It's a slow, progressive, and eventually threshold trigger of metabolism, so that the animal can remain in a very low metabolic state for an extended period of time. Do you want to talk a bit about how you think that triggers in relation to this glycocalyx structure in particular?
Yeah. What we think happens, we observe this, that within the glycocalyx we've got a lot of sodium circulating. There's sodium, chloride, trace elements, everything going on. But as the winter comes on and it gets darker, you slow down, it gets cooler. And sodium sulfate, our glycocalyx is full of connected nanotubes of sulfated hydrocarbons, heparan, sulfate, and so on. Sodium sulfate itself has a very peculiar property that it precipitates out as a solid, just sodium sulfate in water at 32 degrees. It suddenly switches off. Now, the body's normally at about 37 degrees, but as the winter comes on, it cools down. And as it cools down, the sodium sulfate that's circulating starts to precipitate out, and it precipitates out inside these helical coils, sodium connected to the sulfate of your coil. And so it shrinks it down and slows it down, so the passage of electrolyte and other goodies like glucose and so on are slowed right down. So it slows right down until you just
until it sort of shuts off, essentially.
That's
yeah.
right.
But again, again, I'm looking at this sort of connected network across every single cell in the body. If you were able to just literally grab the dimmer switch, right, and you're doing every single part of the body all at the same time at the same rate, that makes sense in terms of how can you actually progressively hit that threshold. Whereas if you're trying to do this, say, cryogenically, for example, there's no way, there's no progressive dial that allows everything to go.
Well, you can freeze people and turn them off.
Sure, but you can't restart them in the same way than hibernation. Yeah. Okay, so Walt Disney is coming back, is that? In theory, so. So it's a very...
It seems to be very simple, and it makes sense.
Yes.
And it's like trees are the same. Potassium sulfate makes flowers grow.
Yes.
Potassium replaces the sodium, and so you switch the plant on. So it's a peculiar property, God's very smart, of sodium sulfate. That's all. The sulfate's there, given in the glycocalyx.
And so would it be fair to say then it's the reason why we don't hibernate versus, other organisms is they've found a way to, the way that their glycocalyx, etc., have been structured is more attuned to be able to do that switch than we are.
They don't have insulation.
So, they've evolved and adapted to that environment. Yeah, but essentially that what you're saying is these channels, because of that dynamic capability, have that ability to, in particular in relation to temperature change, and probably diet around that would have a similar effect potentially as you, for example, you're talking about flowers. You get a change in various conditions, you can actually slowly close these channels down to the point where, okay, now it's time to sit down and take that winter nap essentially.
You've got no choice.
Yeah, that's right, because it's naturally occurring. Yep, okay. The last one I want to talk about then, I know you've also then related that to potentially to things like acupuncture, right? And that this idea in sort of Eastern medicine in particular that we have all these meridians and all these sort of connected channels through the body. Do you want to talk a little bit about how you think acupuncture relates to this glycocalyx structure?
Well, it seems fairly obvious that we've got the glycocalyx, the endothelial surface layer, circular, are filling everywhere in the same way that cell membranes fill the whole thing with tissue and cell. So they're all connected up to the eyes to the end. So there's a conducting matrix that goes everywhere through the body. So we've got all these electrical signals going along with the nerves are propagating on. It's connected to this. So the Chinese acupuncture makes sense that if you put in a needle, you're interrupting
you're perturbing a particular section.
Perturbing the flow, and if the flow is distorted and not working right, if the channels are disconnected and so on, putting in the needle a conducting metal spike will change that, perturb that, and switch it back to normal. So that's what seems to be happening. It makes sense. I mean, it's not just magic. There seems good reason because we've got this entire world of conducting channels that can go wrong with disease. Then if you change the passage of the electrical signals by inserting a needle, then will make some kind of...
And the fact that it's a physical system but dynamic in the sense that, perturbing a particular area, it makes at least logical sense that the ripple of that effect is going to be felt along all of the connected channels to the area, and hence you can sort of stick a needle in your hand and clear up your sinuses as demonstrated consistently with acupuncture. Yeah. Okay, all right. That's a fair bit. I think that might do us for today. Is there anything in particular that you've sort of thought of that we didn't cover along the way in terms of the point of today? I think is to try and sort of cement this, at least in our minds, of this. We have these physical connected channels that can be dynamically altered, either temporarily, semi-permanently, sort of thing, with lots of different things, and those effects can, we hope at least, begin to explain many of the things that are either not very well explained or there's a degree of variability in the explanations or things that they don't sort of explain. Is there anything we've missed, do you think?
Well, I think the whole of biology has missed that. You can't, if you don't know that there's structure to this fuzzy layer that people learn about for yonks. If you don't know there's any structure at all, then enzymes are hanging around somewhere doing what the hell they're doing, but they're stopping and having cups of tea, but then going about their business. But there's nothing, there's no rhyme or reason behind it. There's no organisation. So it seems to be necessary that there's some organisation. It would be rather silly if you have a very intricate organisation of the cell membrane with different channels that you're modelling and this and that and the other and ion pumps, and that you ignore this whole fuzzy layer which is hundreds of times bigger. And well, it's there. Well, that's a huge omission in the whole system. But what our bleating and our going on about things is that once you know that the glycocalyx and the endothelial surface layer have a structure, that it can change, it's dynamic, it's intimately involved in everything, then a whole lot of stuff opens up, which otherwise you're stuck and frustrated. And you can grab a little bit of the thing, the whole picture with enzymes and so on, but you can't get a decent picture. So understanding the structure of the glycocalyx is absolutely crucial. So we think that's really very important indeed.
Yes, as a framework to understand many of these biological reactions.
You can't do anything without it. It's like studying cells and say they don't have a membrane. It doesn't make sense.
All right, well, we'll leave it there today. Thanks. I think that's certainly opened up a lot, hopefully a lot more colour to the story about how this framework of this GCSL area is an explanation to many of the at least limitations in particularly in biological functions. Yeah.
I hope so. Maybe all a big story. Who knows?
All right, we'll leave it there for now. Thanks.